Hello, students. Welcome to my screencast on cellular respiration to help you get ready for your test. Our friend, the mitochondria here. So I took a picture of our fitness room here at TASM, which I hope you're all using. It's a wonderful place. All right, and what's going on with the mitochondria in there? So just like all my videos, this is not for profit. This is mainly meant to help you, only meant to help you students or other students that may benefit from it. The images came from, I tried to be more careful this time and only use images that were licensed for reuse or from Khan Academy. And you can access all Khan Academy items on khanacademy.org, as I know you do for your math class. All right, so let's jump in. I see lots of memes about the mitochondria. People love to post them all over the place. You shared some with me. That was fun. What's the point of it? What does it mean to say powerhouse of the cell? Well, hopefully if you think through it, and as you've learned in this class, it deals something with glucose being a high energy molecule, but we need to break that down into smaller bits of energy that we can use in our bodies. And those smaller bits of energy are called ATP. This is a pretty complicated biochemical pathway. You can see lots of words here, lots of symbols, lots of arrows and directions. So I want you to step back and always see the forest from the trees and see the big picture. We're trying to take glucose and we're trying to make it into a usable energy for us. This is pretty cool. When we talk about it in evolution, etc., we'll see glycolysis all over the place, and then how the mitochondria came to be in eukaryotic cells. But that'll be in a later unit. So let's jump in. Can you think in the big picture, what's the overall formula in words and symbols for cellular respiration? Right? See if you can get it in words first, and then if you can get it in symbols, you're really on top of it. Hopefully you got something like this. Glucose plus oxygen in the process of cellular respiration, or going through the process of it, will yield carbon dioxide, water, and energy, or ATP. Pretty cool. This is the exact opposite of the photosynthesis equation. So you can see why cellular respiration and photosynthesis are a cycle. The products of one are the reactants of the other. Now this will change slightly if there's no oxygen around, but for now, let's go through aerobic respiration or cellular respiration dealing with oxygen. Okay, so what does ATP do in our bodies? What does this molecule do? Well, let's try and use this word bank. There's a couple words over here that I forgot to take out from last year, but we haven't gone over hydrolysis or dehydration synthesis yet. So you can skip those, but see if you can fill in the rest. See if you can figure out what's going on. Hopefully you got something like this. This is from Khan Academy, right? ATP is like a charged battery. So it has energy, like we said earlier. And when we cleave that third phosphate off the end of it, it's going to release some energy. It can go do something. It's like taking, charging up your phone, and then you can go do stuff with it. And then once it's uncharged, we can recharge it. So we don't just throw ADP away. It's ADP stands for 2, ATP stands for 3, right? Tri, and this would be di. And so we can add another phosphate back on, and that's going to happen in this mitochondria. Right? It can also happen in glycolysis, but we'll get more of it in the mitochondria. So hopefully you can see the cycle here, and you're going to see that there's going to be lots of cycles within cellular respiration and within photosynthesis in that video. All right, here's an example of ATP in action, right? So that you can remember that big picture of energy going on. So as this molecule of ATP hits this protein channel, it's going to change the shape. It was closed, and then it becomes open. As it becomes open, it's going to push 3 sodium ions out and bring 2 potassium ions into the cell. This is important for action potential, so how you actually think in your brain. So that's pretty cool. And you use a ton of energy for the sodium potassium pump while you're sleeping and while you're hanging out in my class. Here's another one. So when you're running around, get ready to play basketball and soccer and any other sports that you might play or activities you might do, track, you're going to use ATP to move your muscles. All right, here's our mitochondria. You'll notice, can you label it? Can you remember the different parts, right? So here's the outer membrane. This would be, let me use my cool pointer. This would be the inner membrane. On the very inside is called the matrix. And in between the outer membrane and the inner membrane would be the intermembrane space. So that's pretty cool. It's all logically labeled, right? And the point of it is by having these multiple membranes that we're able to have emergent properties, where we're able to have different pH and enzymes on the inside than on the outside. And that's going to be able to do special things to the molecules as they move around. So if you remember the big picture, we started with glucose on the outside. So can you model and describe glycolysis? Do you remember what's going on there? I'm going to have slides after this one to go into each of these in detail, but can you remember the overall big picture? How about the Krebs cycle? And last but not least, how about the electron transport chain? So if you've got it, and if you can model that and think through this mitochondria here, remember I told you don't need to worry about any of the numbers, but the process, then you're good. You probably don't need to watch the rest of the video except for maybe the fermentation part. All right, so glycolysis yields 2 ATP and 2 NADHs. What does that mean? We're going to take glucose out here. We're going to use enzymes, 10 enzymatic steps to, and we're going to put actually some energy into it to break it. So we're going to break that glucose molecule. It's going to break in half. When it breaks in half, it's going to give you 2 electron carriers. These 2 electron carriers are called NADH. They're going to take those electrons to the electron transport chain. I told you in class they're kind of like empty taxi cabs. They get filled up. They get filled up with 2 electrons and a hydrogen proton each. And it yields a little bit of energy. So this is good. It gives us some energy that we can use. All right, the Krebs cycle is also going to give us some energy. This is happening on the inside in the matrix of the mitochondria. The Krebs cycle is going to take the 2 broken apart glucoses, or they're now called pyruvate. They're going to go to the inside here in the matrix, and there we're going to strip them of their electrons and of their hydrogens, and we're going to make 8 NADHs, or 8 electron carriers, and 2 FADH2s, another type of electron carrier. We'll make 2 molecules of ATP, which is great, and it will also make a bunch of CO2, and that's going to be a waste product. Just like oxygen is a waste product in photosynthesis, carbon dioxide is going to be a waste product in cellular respiration. That's going to diffuse out of the matrix here, out of the mitochondria, out of your cell, and eventually to your bloodstream, and then to your lungs, and then out of your body. And you send it out. All right, then the electron transport chain, last but definitely not least, right? It's going to make lots of ATP. Notice how it makes so much more. So it's really good for us to do. It's going to make water, and then it's going to empty the taxi cabs. And that's what we went over. It's going to give you back the NAD plus and the FAD, so that way you can do glycolysis in the Krebs cycle again. So let's dive in and see each of these happen. And really, this is a good example of how the structure of an organelle leads to its function, right? By having all these folds in here, there's lots of area for it to do the electron transport chain. There's way more than one ATP synthase in here. All right, let's dive in, students. Here is here's a big picture just to remind us. Glycolysis, we're going to split the sugar in half, the glucose. The pyruvate's going to enter into the matrix. It's going to undergo a series of reactions called the citric acid cycle to make lots of electron carriers. They're going to go to this inner membrane in the electron transport chain, where it's going to drop off the electrons. It's going to create a hydrogen proton gradient that will then diffuse through ATP synthase to make ATP. Okay, let's go over glycolysis. What are the inputs and outputs? What's going on here? This is from Khan Academy, and this is from not the AP Khan Academy, but the high school site. It's a little complex. I told you don't need to know any of this stuff. I just want you to get the big picture of what's going on. So here is this glucose molecule. These purple rings just represent carbons, right? So it's going to be C6H12O6. And that's what the plant made for us. So your body's going to take this glucose molecule and in the cytoplasm, that means outside the mitochondria and the fluid inside the cell, there's going to be enzymes there, and it's going to take 2 ATP and those enzymes, and it's going to break this in half. And so it goes through a series of reactions to break the glucose in half. Don't worry about any of these words, to make pyruvate. And so pyruvate is a word you can know. You'll make 2 pyruvates. Here's just featured 1, but you need 2 of them, right? Because there's 6 purple rings here and 3 here. So they're going to be 3 carbon molecules. Along the way, you're going to end up making 4 ATP. So it took 2 in, made 4, so it's going to give you a net gain of 2 ATP. You're also going to take 2 NAD plus and make 2 NADHs. Once again, don't worry about the numbers, but the inputs or the reactants will be glucose, ATP, right? Because it has to go in. You're going to get out ATP as well. NAD plus is going to go in. That's an input, and then you're going to get out NADH, or that is an output. All right, so you can practice that, and we'll practice that in class with our last review sessions. Okay, the Krebs cycle is pretty complicated. It's also called a citric acid cycle because the first step in the process here forms citrate or citric acid. I told you don't need to know about acetyl coenzyme A or the link reaction. If you'd like to learn more about that, you can take AP biology with me. So think of it this way. The pyruvate comes into the matrix, onto the inner part of the mitochondria in here. Along the way, it's been slightly modified and it lost 1 carbon, and that left as carbon dioxide and is now what's called acetyl coenzyme A. That's okay. You don't have to know that or memorize that. So you can think reactants would be pyruvate comes in. Well, let's look what else is a reactant. What else is being used here in the matrix? Well, NAD plus and FAD. All right, and so this process is a cycle because that's more energetically favorable. So the acetyl coenzyme A comes in. It's going to get attached to a pre-existing molecule, this one over here. Don't worry about the names of any of them, but what I told you and what I hope that you can remember, it's important, is that you're taking this pyruvate, you're taking the broken apart glucose, and you're filling up as many taxicabs as possible. You're taking as many of these, let me use my highlighter, NAD pluses, and making NADHs. So you're taking the high energy electrons from glucose and you're taking them and putting them in an electron carrier. You're also taking a hydrogen proton. This releases carbon dioxide as well. Unfortunately, it's covered up here by my little pointer bar. Oh, there we go. You're doing the same thing here at FAD. So you're filling that up to make it FADH2. All right, so what is the point here? The point is to make NADH, FADH2. What went into it? Pyruvate, NAD plus, and FAD. What comes out? NADH, FADH2, and carbon dioxide. All right, let's go to the electron transport chain. Also, a fancy term for this is oxidative phosphorylation. Don't need to know that for this class, but it is in the Khan Academy site, so I put it there. So where are we? Big picture. This on the inside is the matrix. We are now at the intermembrane space. Oh, I'm sorry. I guess the gray would be the matrix here. The intermembrane space in between the very inside of the mitochondria and the outer membrane. Let me go back here. We're at this point, the intermembrane space. What we're doing is NADH and FADH2 come to an electron transport chain, a series of proteins. We did a POGL on this, and I'd highly refer to you to go to that model and to see if you can explain the model. NADH is going to drop off its electrons. So is FADH2. The electrons, remember, are full of energy, and they go along this chain. And as they go past each protein, they pump a H plus or a proton out. So the energy is used to pump H pluses out. The H pluses came from NADH, right? And here's that H plus. The H plus gets dropped off as well. The H pluses get pumped into this intermembrane space. The mitochondria is going to accumulate lots of H pluses in the intermembrane space. Let me show you a big picture. A bunch of H pluses out here. That's going to provide a concentration gradient for them to diffuse. They're going to go from an area of high concentration to low. All right, so they could, because these are membranes, they're impermeable to ions. So they cannot go through this phospholipid bilayer membrane. So the H pluses are stuck. They're like, where do I go? Now, we shouldn't give them human feelings. They're not human molecules, but they're going to diffuse through this protein complex called ATP synthase. And so the H pluses, there's more of them here, less of them here. So they're going to diffuse from an area of high to low, as written right here. And for every 3 to go through ATP synthase, it's going to turn the rotor, and it phosphorylates an ADP, adding a phosphate to make ATP. In other words, it takes 2 phosphates, it adds the third, and it makes it into ATP. How cool. So it has made energy for us to use. It took the empty batteries and it charged them up. This can generate lots of ATP, way more than glycolysis or the Krebs cycle, but only if the electrons can keep going through the chain. So what do the electrons do at the end of the chain? How do they get off of the highway? And I kept talking about like salt and caboose highway. How did it get off? So the off ramp is the electrons are going to be accepted by oxygen and the hydrogen protons to make water. So oxygen is electronegative. It's going to accept the electrons and a hydrogen proton, and it will form water. If there's no oxygen, then this is going to shut down. So that's why it's so important to have oxygen to do aerobic respiration. All right, let's wrap this up. You just heard the bell, so I'm going to need to get going. Here we just did oxygen present, we did the Krebs cycle, we did oxidative phosphorylation, or the electron transport chain. That made a lot of ATP. Well, what if there's no oxygen? What if you're running around and you run out of oxygen, or you're swimming, or you're playing a sport, or what if it's yeast like in your lab that you did and they run out of oxygen to do? Do they just die? Does it just stop? No, hopefully it will go through anaerobic or anaerobic respiration, or it will go through fermentation. And so there it's going to do a different process. It's going to continue to do glycolysis and try and produce ATP, but it will not do the electron transport chain. It won't do the end product there. All right. There is something called anaerobic respiration where some organisms can use something besides oxygen as the final electron acceptor. Don't worry about that as much. Let's just think oxygen, okay, we can do the electron transport chain, no oxygen, fermentation. So let's see fermentation in action. You did a lab on it. You did a claim evidence reasoning, so I feel pretty good about your knowledge about this. I was excited about your labs. Sorry, it's the same picture here. So no oxygen, so this is going to shut down. So without oxygen, it cannot do the electron transport chain. It also can't do the Krebs cycle because there's no NAD plus to put the electrons into. So we're just going to stick with glycolysis. So your body's going to take, this is happening in you and in some bacteria, this is lactic acid. Your body's going to take the glucose and is going to split it. You then have pyruvate. This makes some energy. But in order for this to keep going on, you need to regenerate NAD plus. You need those empty taxicabs. Without them, you can't keep doing glycolysis. Eventually, you'll run out of NAD pluses to put the electrons and the hydrogen in as you're splitting the glucose. You can't just split glucose into pyruvate without making NADH. It's impossible. So you have to have some way to have the reactant be regenerated. In other words, you have to be able to get the electrons and the hydrogens out of NADH so that you can have an empty taxicab. So how does your body do that? It's able to use an enzyme to take those electrons and hydrogen, put them onto pyruvate, which is going to change it into lactic acid. When you add the electrons and hydrogen onto pyruvate, it is no longer pyruvate. It becomes lactic acid. You'll incur an oxygen debt, and after a while, eventually you'll catch up on oxygen. You'll be able to move that lactic acid to your liver and process it. That's what happens in you. In yeast, they're going to do a different process where they're going to use 2 enzymes, not just one, to make ethanol. Sorry, my head is covering it up here. I don't know if I can move my head. Well, okay, we're almost at the end of the video. So the glucose is going to be split. And then pyruvate is going to be turned into acetaldehyde with this enzyme, pyruvate decarboxylase. You don't need to know the enzyme name. This is going to release carbon dioxide, and this is what you actually measured in your claim evidence reasoning, right? You measured it with the height of the foam. Acetaldehyde can then be rearranged into ethanol with this enzyme, alcohol dehydrogenase, and this is going to regenerate NAD, or it's going to remake the NAD+. It's going to strip the electrons from NADH and the hydrogen proton. It's going to make NAD+. So can you think big picture? How do we get energy? Sorry, I'm going all the way back. When we don't have oxygen, right? And so if we go all the way through here to the end, you keep doing glycolysis, and to do that, you have to keep regenerating NAD, which is going to make lactic acid. So I thought this flowchart from Khan Academy is very good, right? So no oxygen, animals are going to do lactic acid. Yeast and some bacteria can make ethanol. All right, it's good that you don't make ethanol. Otherwise, you would be intoxicated every time you ran around. All right, so why is this important? Actually, just today in class, right, we researched mitochondrial diseases. We learned about Leigh syndrome and some of the sad illnesses that can happen with mitochondrial defects. And then we'll talk a little bit about cyanide and some creatine supplements. So I hope that you enjoyed this, and good luck for your test on cellular respiration.